I see a light.
A light?
Yeah, over there. Hey, Conscience, am I dead?
No, I see it too. What is it?
It's so pretty.
I'm feeling happy, which is a big deal for me.
I want to touch it.
Ooh. Hey, come back. Come on back here.
I'm gonna get you.
I'm gonna get you.
I'm gonna get you.
I'm gonna swim with you.
I'm gonna get you.
I'm gonna be your best friend. Good feelings gone.
In this clip of Finding Nemo, it shows a perfect example of how an anglerfish uses its natural light to attract prey for food. The anglerfish and many other deep-sea animals who live in complete darkness rely on their bioluminescence to survive, and some have certain uses for them. Many of them simply use their light to help them navigate through the darkness, such as these jellyfish. Although many bioluminescent things occur in the deep sea, there's actually some that we can see on the ocean shore. For example, like this glowing sand, or the crashing waves that appear to be making light. But what makes them glow is actually tiny planktons called dinoflagellants who have their own bioluminescence. They light up when there's agitation or sudden movement. And their purpose of making light is not to look pretty, but to act as a distraction for when there's an oncoming predator. Some even use it for self-defense, like this cardinal fish. This fish ate an ostracod which has bioluminescence in it. Once a predator is near, the fish will spit out the ostracod, causing it to glow and leaving the predator frightened, which also allows the fish enough time to swim away. Did you know that these glowing planktons were actually pretty useful for the Japanese armies during World War II? Well, the Japanese armies would go to the beach and fill up a jar of plankton. Then they would give it a little shake. And with that light, they would use it to read maps at night. Bioluminescence is still pretty useful to this day. For example, some are studying how to track subs and torpedoes with bioluminescence. With all these beneficial uses, you may be wondering how bioluminescence was discovered. For a very long time, bioluminescence has been a mystery in science up until the 1960s. At Nagoya University, a scientist named Osamu Shimomura made a huge discovery from an ostracod. With the ostracod, he was able to extract luciferin, which has never been done before. In this photo, Shimomura is crystallizing the luciferin, which helped him better understand the structure of the molecule. Upon further research, he also discovers that luciferase is an enzyme that catalyzes the reaction between luciferin and oxygen, which causes the glow. Although most bioluminescent creatures have luciferin, there are some exceptions where some animals have green fluorescent protein, also known as GFP. In this situation, blue light is absorbed by GFP, which gives off a green light. Before I explain how these reactions work, I want to take note that bioluminescence includes a concept called chemiluminescence. Chemiluminescence is a chemical reaction that excites a molecule to a higher energy state, and once it loses energy and returns to ground state, it emits light. To give a visual representation of how chemiluminescence works, I'll be using an atom instead of a molecule. When an atom is excited, it moves on to the second energy level. Then once it loses its energy and moves back to ground state, it emits a light. This is the basics of chemiluminescence, but it's more complex because it deals with the molecule. A great example of chemiluminescence would be a glow stick. When you crack a glow stick, the chemicals inside mix together and produce a light. However, unlike a glow stick, the light in bioluminescence does not fade away. Depending on which animal you're looking at, reactions will differ. However, in almost all reactions, you will always find luciferin, which produces the light, luciferase, an enzyme that catalyzes the reaction, and oxygen. How this reaction works is first luciferin and luciferase combine to make a complex molecule. Then it undergoes a reaction with oxygen, making oxyluciferin. And with that molecule, they excite it to a higher energy state. And once it loses its energy, it can emit light of varying colors, depending on the light's wavelength. If we were to look at one bioluminescent animal, like a firefly, this is how the reactions would look like. It would first start off with arbutin and combine with hydrolase and oxidase. Then that molecule would also combine with other molecules and finally end up with D-luciferin. D-luciferin will then combine with ATP and ionic magnesium, and luciferase will catalyze the reaction to make a complex molecule. Under the reaction with oxygen, the bonds will move around and create oxyluciferin, which in turn creates two types of molecules which will either release a yellow or red light. What's so unique about GFP is that it actually doesn't need any additional molecules and can glow on its own. The shape of GFP can be seen as a soda can. Around it are 11 beta sheets which are connected to the amino loops at the end. In the center is a molecule called chromophore containing amino acids that make it glow, which are serine, tyrosine, and glycine. GFP can fold on its own and is non-toxic, which is why it has been widely used in cells. GFP can only emit one light, which is green. So for experimental purposes, scientists mutated the gene to change the color. So now we have M. cerulean, EGFP, MKO, M. cherry, and M. plum. Because they were able to do this, it was very revolutionary in biology. Since GFP has different colors, they have been used to help identify different parts of the cell. Scientists have also used the same concept for brains so we can learn more about it. And GFP has also been used in cells who have been genetically modified to help regulate it. In biology, they've used bioluminescence to detect invisible genes and proteins in cells. Although our technology today is advanced, scientists haven't really made a close match with bioluminescence. However, they're studying whether or not it can be genetically modified with plants and animals. The Navy is also developing an aquatic robot that can measure bioluminescence to detect enemies and prevent an accidental reveal. With the help of Shimomura's discoveries, many other researchers have begun to experiment with bioluminescence. Once scientists get familiar with bioluminescence, they're planning on genetically modifying trees to make them glow. They hope that by doing this they would be able to replace streetlights. If this were possible, it would save money, electricity, and even be healthy to our environment. Also, bioluminescence is considered a cold light, so glowing trees wouldn't be a safety hazard. Another way scientists are using bioluminescence and GFP is by inserting it into genes to possibly cure viruses. An American-Japanese scientist inserted green fluorescent protein within cats' genes. The purpose of inserting these proteins was to help protect them against feline immunodeficiency virus, which is FIV and is the human equivalent to AIDS. This study has proven to work, but because there is GFP in their genes, these cats glow at night, so there is still more work to be done. However, once they find a way to get rid of the glow, this could be a possible solution to curing AIDS. Another use of this can be used to help observe and analyze cancer. In this photo, it shows a mouse's cell that has a brain tumor. The mouse's blood vessels are seen through green GFP, and the DS red expresses the brain tumor of the mouse. This has helped many scientists understand tumors. For this demo, I used a highlighter, some water, and a black light. To prep for this demo, I first took this highlighter and popped the bottom of it off to get to the ink. Then I took the highlighter ink and placed it inside the water. I left it there to soak for about an hour or so. Now that the highlighter dye has been soaked completely, I took it out and threw it away. Now to continue on, I'll turn off all the lights. and as I'm doing so, I'll explain what's going to happen. So the highlighter dye is supposed to represent as luciferin, which makes the light, and the black light is supposed to represent as luciferase. So once I turn on the black light and put it over the highlighter dye, it should glow. Once the lights are off, you can see that the highlighter dye is not exactly glowing. So now that I've turned on the black light, the water has glown. Since this demo was pretty short, here's another clip of someone making glowing light out of different materials. The key material in this experiment is luminol, which produces light. First, he measures out 0.2 grams of luminol. Then he adds 12 grams of baking soda and half a gram of copper sulfate. Then he pours in half a liter of water. And then he pours in ammonium and sodium hydroxide solution. Then finally he mixes them all together. In the other solution, he pours in 5 milliliters of hydrogen peroxide. Then he dilutes the solution by adding half a liter of water. Then he pours in half a liter of each solution into a measuring cup. He turns off the light and mixes them together to get this glow. Now for some interesting facts of how people experimented with bioluminescence. A North Carolina-based company is planning to release glow-in-the-dark candy. It's made from the same enzymes as found in bioluminescent animals. Once you lick it, it should glow and should last between several seconds to minutes. They're also wanting to add this to beverages and edibles. However, they're still waiting to be FDA approved. Plant biologists in the mid-1980s created a glowing tobacco plant using firefly bioluminescence. No one ate this tobacco plant, and it began a new way to study plant cell biology. Plant biologists to this day are still using this concept. Around the early 2000s, Anthony Trewavas experimented with bioluminescence in plants. He made a fluorescent potato that glowed under a black light when it was dehydrated. This interested farmers because it could tell them how to take care of their crops or what was wrong with it. However, because it dealt with genetically modified plants, it wasn't a very huge success in Europe. Even though GFP was originally found in jellyfish, or more specifically, the Echorea victoria, there have been many uses for them and have been used in all types of animals and plants. It has helped advance biomedicine and researches that have solved many problems. Learning about bioluminescence will not only help us understand the thousands of deep-sea animals, but could also be very beneficial for our technology and can offer endless possibilities.